T-cell biology is complex, and in this module we will provide a summary of the major processes and events that unfold upon T-cell stimulation. Following antigen exposure, stimulated T-cells undergo clonal expansion and differentiate into effector cells that manifest effector function through cell-mediated cytotoxicity or cytokine secretion. In order to proliferate, differentiate, and express effector function, activated T-cells undergo a significant shift in phenotype compared to their naive counterparts. This shift involves the induction of a myriad of factors, including effector cytokines, lineage-restricted transcription factors, and surface markers listed here. CD4-positive T helper TH cells play a critical role in coordinating almost every aspect of the adaptive immune response and are described as the generals of the immune system. T helper cells fulfill this role mainly through the secretion of cytokines. Notably, T helper cells stimulate cellular immunity by activating cytotoxic T cells to kill infected or transformed cells, and stimulate humoral immunity by potentiating antibody production by B cells. CD4-positive T helper cells also stimulate nonspecific or innate immune responses by, for example, stimulating macrophages to destroy ingested microbes. Naive CD4-positive cells are activated by antigens presented by antigen-presenting cells. Depending on the intensity of stimulation, patterns of cytokine exposure, and other factors, CD4-positive T cells differentiate into different subpopulations with distinct functions and properties. Th1 T helper 1 cells are induced when naive CD4-positive cells are activated in the presence of gamma interferon and IL-2. These cells play a key role in potentiating cell-mediated immunity and inflammation. Th1 cells have an important role in the elimination of intracellular pathogens and in potentiating anti-tumor responses. Th2 T helper 2 cells are generated in the presence of IL-4, enhance antibody-mediated immunity, and have an important role in responding to extracellular parasites. Th2 cells also secrete cytokines that repress Th1 cell responses. Regulatory T cells, or Tregs, are generated in the presence of IL-2 and TGF-beta and serve to maintain tolerance to self-antigens and suppress immune responses, thereby preventing the development of autoimmune disease. These cells also suppress effective anti-tumor responses. Th17 cells are generated in the presence of IL-6 and TGF-beta and have been implicated in the immunity to extracellular bacteria and fungi. TFH, or follicular helper cells, are generated in the presence of IL-6 and IL-21. These cells promote humoral immunity by supporting B cell development. Note that this list of T helper subsets is not exhaustive. Others have been described, including Th9 and Th22 cells. The ability of different cytokine milieux to control T helper cell fate and effector function has been attributed to the selection activation of STAT signaling proteins and the subsequent expression of select master regulators or lineage-specific transcription factors. Th1 cells are, for example, produced as a result of STAT4 activation by IL-2 and the subsequent expression of the T-box transcription factor Tbet. Tbet is a master regulator of Th1 differentiation, and together with STAT4, promotes the expression of the stereotypic Th1 cytokines such as interferon gamma. Th2 lineage differentiation and the production of signature Th2 cytokines such as IL-13 and IL-4 results from the activation of STAT6 and the subsequent expression of the master regulator GATA3. Th17 cells and associated cytokines IL-17A, IL-17F, and IL-22 are produced as a result of STAT3 activation and expression of the transcription factor ROR gamma T. TFH cell differentiation and cytokine production are dependent upon STAT3 signaling and expression of the transcriptional repressor BCL6, while the production of Treg cells requires STAT5 signaling and expression of the transcription factor FOXP3. The expression of lineage-specific transcription factors and signature cytokines provides for sets of markers that can reliably be used to identify and differentiate the different T helper subsets.